and welcome to episode 16. In the last few episodes, I've been discussing DNA, chromosomes, genes, and alleles. Last episode, I specifically talked about transcription and translation, which was dotted with a few explanations on the structure of various parts of the DNA molecule. In today's episode, I'm going to be discussing how DNA is produced, or synthesized, and how it's repaired. Naturally, any discussion on DNA repair has to involve a little talk about DNA damage, specifically mutations. I'll end this episode by talking about the different kinds of mutations and what they mean for the organism whose DNA suffered that mutation. With that said, let's dive right into it. Deoxyribonucleic acid is composed of two strands, wrapping together in a helix. Each strand in the helix is complementary to the other. This is to say that they're each lined with a series of nucleotide bases, A, T, C, or G, and these bases interlock in pairs to create complementary strands. The A and T bases, or adenine and thymine, are able to pair together. Similarly, the C and G bases, or cytosine and guanine, also pair together. When one strand has a sequence like ATACCG, the other strand has a complementary sequence that goes something like TATGGC. This is called complementary base pairing. It's easy to remember because it's pretty self-explanatory. Recall the directionality of DNA that I described in episode 4. The ribose sugars in the DNA backbone are connected through phosphate groups that link them together, binding to the 5' carbon of one sugar and the 3' carbon of the next. As the ribose sugar polymer forms a strand, it has a 3' end, where a hydroxyl group on the 3' carbon is exposed, and it also has a 5' end, where a phosphate group dangles off the end of the 5' carbon. I should also mention that DNA is anti-parallel. This means that the DNA strands run in opposite directions from one another. If you were to take a strand of DNA and lay it down in a straight line with one end on your left and the other end on your right, each end of the DNA molecule would have both a 5' and a 3' end, one from each of the strands. If you were to split the DNA into separate strands, one strand would have its 3' end by your left and its 5' end by your right, while the other strand would be oriented in the opposite direction. During DNA synthesis, new nucleotide bases are only added to the 3' end. This makes some aspects of DNA replication relatively complicated. I talked about DNA replication a little bit during the S phase of the cell cycle, but now I'm going to go into much more detail. DNA displays a semi-conservative replication. This means that during replication, one molecule of DNA is split into its two component strands. Each strand then serves as a template for the creation of a whole new complementary strand of DNA. When synthesis of the complementary strand is complete, we now have two new molecules of DNA. The DNA has been replicated. This is semi-conservative because each daughter DNA molecule has one strand from the parent DNA molecule and one new strand that was recently synthesized from scratch out of the nucleotide bases in the solution. The actual process of DNA replication involves a large suite of enzymes and protein complexes. The enzyme that actually adds new nucleotide bases to a newly forming strand is called DNA polymerase. Nucleotides are only added on the 3' end because that's really the only way that DNA polymerase can attach new bases. The enzyme can only interact with the reactive hydroxyl group on the 3' end. DNA polymerase just doesn't have the molecular machinery to interact in a sufficient way with the phosphate group on the 5' end. Because of this, as DNA polymerase does its job, it slides down the newly forming DNA strand in a 5' to 3' direction. These new bases are added through phosphorylation reactions by molecules called DNTPs. Recall the molecule ATP. It stands for adenosine triphosphate. You might have noticed that adenosine is also a nucleotide base. If you listened to earlier episodes, you might have heard me mention GTP, or guanine triphosphate. This is a similar energy-carrying molecule that uses the nucleotide guanine instead of adenine. When a nucleotide base from DNA is bound to a triphosphate group, it becomes a DNTP. Have you guessed what DNTP stands for yet? It means deoxyribonucleoside triphosphate, because it uses nucleotides derived from and used for deoxyribonucleic acid. Okay, so now that you know the enzyme that adds the nucleotides, the direction it works in, and a little bit about the DNTPs, let's get into the mechanisms of replication itself. The replication of DNA begins at a place called the origin of replication. This origin of replication is where the DNA strands first split apart. Bacteria only have one origin of replication in their DNA, but eukaryotes can have many. When the DNA strands split at the origin of replication, a suite of proteins facilitates the split and allows it to continue. The nucleotide bases in both directions from the origin begin to unzip. Their hydrogen bonds are broken. A bubble emerges, a bubble formed by the growing space between the two strands. At each corner of this almond-shaped bubble, the DNA splits from one strand into two strands. These corner points are called replication forks, as their Y shape resembles a fork in the road. Recall the suite of proteins that I mentioned a moment ago. These help open up the DNA at the origin of replication and propagate the process. The first of these proteins is called DNA helicase. This protein binds to the origin and breaks the hydrogen bonds between nucleotide bases. This initiates the breakage and begins the formation of the replication bubble. A group of proteins called single-strand DNA binding proteins, or SSBPs, come along to help the DNA helicase do its job. You see, if the DNA strands are cleaved apart, but then just ignored, if they're just left to float around, they'll rapidly reattach to one another. The complementary hydrogen bonds will attract one another, and they'll just zip right back up. If this happens, the DNA helicase will not really be doing anything remotely productive. It'll just be running down the length of the DNA, unzipping the bases in front of it, only for the DNA to naturally zip itself back up right behind it. These SSBPs prevent this from happening. As their name suggests, they bind to one of the two separated strands, and this keeps them from reassociating with one another. This keeps the replication fork open, so that the bubble can continue to expand. As the DNA helicase works its way down a strand of DNA, unzipping the bases of the replication fork, it creates tension in the unzipped portion of the DNA. If you have a strand of yarn, you'll notice that it's made of smaller strings, all wound together. If you grab the tips of those small strings and pull, in an attempt to unravel the strand of yarn, you'll quickly cause that strand to coil up with tension. DNA really isn't that much different. As the DNA helicase unzips the DNA and the SSBPs pull each strand apart, a tension is induced in the unzipped portion of the DNA. So an enzyme called topoisomerase moves ahead of the DNA helicase, and it makes small cuts into the DNA to relieve the tension, and then they reseal these cuts before the DNA helicase gets to it. In this way, the topoisomerase is kind of like a pressure gauge that lets out some steam when the pressure gets too high. When the DNA gets too tightly wound up, the topoisomerase makes a few snips and cuts here and there, relieves the tension, and then patches everything back up and moves along. So now that the DNA is opened up, it's time to start synthesizing the new strand. Recall that DNA is only synthesized in the 5' to 3' direction, as DNA polymerase can only add bases to the 3' end. DNA polymerase also needs a base with an exposed 3' end to start off of. It can't lay down its own track, so to speak. It needs a starting point. It needs somewhere to bind to so that it can set up camp and kind of get started. So where does the DNA polymerase start if the replication bubble only has two single-sided strands of DNA? It starts with a small piece of RNA called a primer. This primer is small, typically 3 to 15 bases long, give or take. An enzyme called RNA polymerase finds the origin of replication and binds to it. As the replication bubble is quite large, the origin of replication is in the middle of each single strand. So this RNA polymerase goes out to the middle of the strand and associates with the starting sequence. Unlike DNA polymerase, RNA polymerase doesn't need a 3' end to start synthesizing its product. So the RNA polymerase builds a short strand of RNA, the primer, right at the origin of replication. This primer provides the 3' end that the DNA polymerase needs in order to start synthesizing new DNA. The DNA polymerase begins, moving in the 5' to 3' direction. This newly created strand of DNA is called the leading strand, as its synthesis is smooth and continuous. As more nucleotide bases are added to the 3' end, the 3' end approaches the replication fork. But the replication fork keeps moving downstream as the bases are unzipped by DNA helicase. The 3' end of the newly synthesized strand won't ever really catch up to the replication fork, so the leading strand is synthesized continuously in a direct and very simple manner. Let's say you're sitting on top of the DNA helicase as it unzips the DNA, and you're facing backwards towards the big open replication bubble. Single strands of unzipped DNA project out to your left and to your right. To your right, you see the DNA polymerase of the leading strand. It's chugging right along behind you in the DNA helicase, building that new strand of DNA, the leading strand. It's being built in one continuous effort. But now look to your left. What would you see over here? Because DNA is anti-parallel, this strand runs in the opposite direction from the strand on the right. DNA polymerase can't chug along right behind you, as it can only add nucleotide bases to the 3' end. It'll be moving away from you in the DNA helicase as it synthesizes its new strand. This new strand is called the lagging strand, or the discontinuous strand, because it has to be synthesized in discrete chunks. Basically, the RNA polymerase has to come and make a new primer at some point along the strand. DNA polymerase associates with the primer and begins synthesis in the 5' to 3' direction. Eventually, the DNA polymerase will come to the middle of the replication bubble, to the origin of replication, where it runs into the first few nucleotides of the leading strand going in the other direction. The DNA polymerase detaches after having synthesized a small stretch of new DNA. The RNA polymerase then goes to a point further down the strand and makes a new primer, and the process repeats itself. The DNA polymerase associates, makes a short stretch of new DNA, then stops when it bumps into the end of the previous DNA segment it just made. These short segments are called Okazaki fragments, named after the researcher whose team discovered them. RNA polymerase makes a new primer, and DNA polymerase makes a new segment. This process repeats in these discrete steps, building the lagging DNA strand, one Okazaki fragment at a time. An enzyme called DNA ligase comes through and seals all the breaks between the Okazaki fragments, smoothing them into a continuous strand of new DNA. So let's go back to that metaphor, where you're riding on the DNA helicase and looking backwards at the replication bubble as it expands. To your right, you see the DNA polymerase of the leading strand following close behind you. To your left, you see RNA and DNA polymerase doing a back-and-forth shuffle as they synthesize the Okazaki fragments. DNA ligase lingers behind them both, smoothing out the fragment sections into a single continuous strand of new DNA. Basically, if you look behind you, in the direction that the DNA helicase is going, you'll see the double-stranded parent DNA that has yet to be unzipped. Now when you turn back around and face to look in front of you, you'll see two single strands of DNA going away to your left and right, with each new complementary strand of DNA being built onto them. This creates two new daughter molecules, with each one strand from their parent DNA molecule. As the replication forks go down each length of the DNA molecule, they'll eventually reach the ends. Basically, at this point, the DNA molecule has been fully unzipped. But as the DNA polymerase is working behind the DNA helicase, by the time the full DNA molecule gets unzipped, you already have each strand almost half finished with their new complementary strand. Ultimately, this creates two new daughter DNA molecules, each with one strand from their parent DNA molecule. You've probably realized by now that this is the semi-conservative nature of DNA replication in action. So that sounds pretty easy, right? Open up the DNA at the origin, make the replication bubble, build your leading and lagging strands, and you're set, right? Well, not quite. You see, DNA polymerase requires a primer to start synthesis. When a primer for an Okazaki fragment is not needed anymore, an enzyme eats it away. The next Okazaki fragment will go right up to the start of the previous fragment, kind of overriding where the primer used to be. When the primer is on the end of a DNA molecule, it'll be chewed away like normal, but this time there's no place for a new primer to attach. DNA polymerase will be unable to fill in the space where the primer used to be. Where each leading strand is a full replication of the template strand. Each lagging strand replicates the template strand with the exception of this last little tail end of DNA where the primer used to be, as well as any stretch of DNA that dangles past it. That small stretch of DNA on the end of the template strand can't get replicated. You might be thinking, so what's the big deal? It's just a few nucleotide bases, maybe 50 to 100 bases, but the DNA molecule is billions of bases long. So how can a loss of 100 bases really even matter that much? The problem is that this happens every single time DNA is replicated. Every single time, a small bit on the end doesn't get replicated, and the strand of DNA shrinks by a little bit. When this strand is itself replicated, its daughter strand will be even shorter, and that one's daughter strand shorter still. As time goes on and the DNA continues to replicate over and over and over again, this little bit will add up, and soon the DNA molecule is suffering serious degradation at its tips. When your DNA degrades, your cells in your body degrade. Our bodies try to prevent this genetic degradation with structures called telomeres. If a strand of DNA could be compared to a shoelace, then the telomeres would be like the aglets, which keep the shoelace from fraying and falling apart. Telomeres are composed of repeating sequences of regular DNA, except they don't code for anything. They're not genes. In humans, for example, the telomeres are composed of thousands and thousands of repetitions of the sequence TTAGGG. An enzyme called telomerase is responsible for building telomeres. Telomerase is the kind of enzyme that isn't strictly a protein. Much like the ribosome, the telomerase enzyme is mostly protein, but also contains subunits composed of RNA. Telomerase uses its RNA component to bind to the bases on the end of the template strand. From here, the telomerase adds bases to the template strand. Not a new strand, but to the parent strand, the template strand, and extends it a little bit. By extending the telomere, it creates another site for the RNA polymerase to create a primer, which allows DNA polymerase to finish synthesizing the end of the gene. It's important to understand that this doesn't actually stop the little bit of DNA from not getting replicated. That still happens. It's just that with the help of telomerase, the template strand can get expanded well past the limit of any actual genes. When the lagging strand is synthesized, the little bit at the end that doesn't get replicated is just part of the telomere. It isn't part of any important gene or protein coding region or some kind of regulatory gene. If those more important regions get damaged, their function would be impaired. In humans, telomerase is active only in the cells that produce gametes. That is, the telomerase only actively works to rebuild and rejuvenate our telomeres in the cells that produce sperm and eggs. Every other cell in our bodies, like our skin cells and our lung cells and our brain cells, are called somatic cells. Unlike the germ cells that make sperm and eggs, somatic cells do not undergo meiosis, they only go through mitosis. On that note, somatic cells only go through mitosis a limited number of times. As somatic cells don't possess telomerase, their telomeres get degraded and they don't get repaired. Once the telomeres are gone, the DNA itself starts degrading. Instead of continually replicating with DNA of worse and worse quality, the cell reaches a point called senescence, where it simply stops dividing entirely. The cell basically decides it can no longer pass on its sufficiently degraded DNA and stops reproducing. One of the leading theories on aging posits that we age because of our cells going into senescence. When you're a newborn infant, your cells haven't had time to replicate that much. Your telomeres are long, and your DNA is healthy, and your cells reproduce with vigor. As you age, your telomeres slowly get degraded, and your cells eventually go into senescence. It's slow at first, not even really noticeable compared to the rate of cells still dividing and making new, healthy daughter cells. But as time goes on, more and more cells will go into senescence. You find your skin starts to wrinkle and droop as the cells who once maintained the collagen are going into retirement. You begin to get forgetful and confused as brain cells go into senescence and shrivel away. Your vision fades as the cells in your eyes follow the same fate, and so it goes throughout your whole body until you reach a critical point where your cells simply aren't dividing in the numbers necessary to sustain your life. And then you die, having hopefully lived a long and experience-filled life. Naturally, research into cures or treatments for aging have put intense focus on telomeres and telomerase, specifically into finding ways to activate telomerase in our somatic cells and not just in the germ cells that produce gametes. Cancerous somatic cells, on the other hand, don't have proper regulatory control over their own replication. Because of this, most cancer cells are actually able to express the genes that produce telomerase. Cancer cells can protect and maintain their telomeres, while your healthy cells suffer the ravages of time and age. For a cell to become cancerous, it has to have some kind of error, or mutation, somewhere in its DNA that disables the regulatory mechanisms involved in cellular replication. Not all mutations cause cancers, but they can impair other functions of the cell. Furthermore, not all errors in the DNA are due to mutation. Some errors, for example, are from mistakes made during the process of DNA replication. Despite working at rates of up to 500 bases a second, DNA polymerase rarely makes mistakes. In fact, less than 1 in 100,000 nucleotide bases organized by DNA polymerase fails to match its associated base on the template strand. How can DNA replication be so precise? And when this replication fails and creates an error, how does that get fixed? First, understand that nucleotide bases don't pair up the way they do for no reason. A and T pair together, and C and G pair together, because these conformations are the most stable. This means that these pairs of nucleotide bases are in their most energetically favorable conformations. When synthesizing a new strand, DNA polymerase is unlikely to make a mismatched base pair, as it would mean doing something that wasn't energetically favorable. It would be a very noticeable bump in the road from the DNA polymerase's perspective. If the DNA polymerase encounters one of these bumps in the road, one of these mismatched base pairs, it immediately halts everything in order to remove the offending nucleotide base. After removing the mismatched base, the DNA polymerase continues to synthesize more DNA. Despite its best efforts, the DNA polymerase can still skip over a mismatched pair from time to time. When this happens, a suite of proteins jumps into play to fix the overlooked mismatch. As DNA polymerase makes about one mistake for every 100,000 bases, the assistance of these proteins reduces the error rate for DNA down to about 1 in a billion bases. DNA doesn't just suffer problems from mistakes made during replication and proofreading. DNA can be harmed and mutated by all sorts of environmental inputs, like cigarette smoking, exposure to industrial chemicals, ultraviolet radiation, and more. Typically, our bodies employ various enzymes that work through various mechanisms to repair this daily damage. Otherwise, the mutations would build up rapidly and kill us quicker than senescence. One such mechanism for DNA repair is called nucleotide excision. When ultraviolet radiation strikes a molecule of DNA, its energy can cause two adjacent pyrimidines to bind together into a dimer. So if UV radiation hits two adjacent cytosines, it can fuse them together to make a cytosine dimer. Similarly, if UV radiation hits two adjacent thymines, it can fuse them together to make a thymine dimer. These dimers are like awkward kinks in the DNA that screw up its structure and impair the function of enzymes and proteins that slide along the DNA molecule. To mitigate this, the cell determines which segment of the two strands of the DNA is damaged. That segment of the strand is removed and destroyed, and then gets replaced by DNA built off of the template of the remaining strand. So I've mentioned mutations many times now, especially in the context of cancer cells, but I haven't yet gotten around to explaining what mutations are. To put it technically, a mutation is a permanent alteration to an organism's DNA. In worst cases, this mutation gets passed on to its offspring. In this way, mutations are more than the mundane UV damage inflicted and repaired in a day's time. Mutations are permanent alterations to the genome, the creation of new alleles that code for new traits. Unfortunately, the vast majority of mutations are harmful, as they disable, already well-functioning enzymatic machinery. So the so-called new traits are something like a less effective lung tissue, or a malformed brain or eye, or some kind of mental retardation. Mutations can affect a single nucleotide base, or they can affect multiple bases. Those mutations that affect a single base are called point mutations. Let's say we have a gene with the sequence TC-TC-AG. This would produce an mRNA molecule with the sequence AG-AG-UC. Real genes are hundreds if not thousands of bases long, but this is just a simple example, so bear with me. Let's say that the third base in this mRNA, an adenosine, is removed in a point mutation and replaced by a cytosine. Now the gene sequence goes AGC-GUC. If you watched the last episode about transcription and translation, you know that groups of 3 sequential nucleotide bases code for specific amino acids, and these amino acids get added to a polypeptide chain that becomes a protein. The type of protein is determined by its amino acid sequences, and the amino acids are determined by the 3 bases, or the codon, that codes for it. I discussed how most amino acids have 2 or 3 different codons that code for them. This is kind of like a built-in redundancy to protect against mutations. Let's go back to our gene sequence for a moment. The original sequence on the mRNA was AG-AG-UC, but the point mutation turned it into AG-CG-UC. If AGA is our first codon, and GUC is our second codon, we can see that our first codon has suffered the mutation to its third base. The first codon, AGA, codes for the amino acid arginine, and the second codon, GTC, codes for valine. But now that AGA has become AGU, it doesn't code for arginine anymore. Instead, it codes for an amino acid called serine. The original gene would have coded for a polypeptide composed of arginine and valine monomers, but the mutated gene coded for a polypeptide with serine and valine polymers instead. The protein is new, it's a product of mutation. In most cases, like 99.999% of cases, this mutation produces a non-functional protein, or worse, an actively harmful protein. But in those extremely rare 0.0001% of instances, the mutation produces a protein that works better than the original, or it provides a novel function that the original protein doesn't. If this novel function provides a fitness advantage for the organism, it'll be more likely to survive and reproduce, passing on this mutant gene to its offspring. If the mutation increased the parent's fitness, it'll likely increase the fitness of the offspring as well. In this way, the mutation will persist in the gene pool, spreading in popularity as it outcompetes and outbreeds those with the inferior allele. In the example I gave with that hypothetical gene sequence, the point mutation in question is also called a missense mutation. A missense mutation causes a change to the amino acid sequence of a protein. A silent mutation, on the other hand, is a mutation that doesn't change the amino acid sequence of a protein. Consider the codons GGU and GGC. Both of these codons code for an amino acid called glycine. If a GGU sequence were to undergo a point mutation to become GGC, it would be a silent mutation, as it did not change the encoded amino acid. If the encoded amino acid isn't changed, the protein isn't changed. If the protein isn't changed, there's really no problem. The silent mutation is silent. It doesn't cause problems. It minds its own business. It stays out of the way. Other mutations don't involve changing one base into another. Some mutations involve the wholesale addition or subtraction of bases from the gene. The addition or subtraction of a single nucleotide base can ruin an entire protein. As the bases in the genetic code are read in groups of 3, adding or subtracting a single base will shift the entire frame of reference and totally scramble the encoded message. Naturally, these are called frame shift mutations. Let's use our original gene sequence again, the AGAGUC, to explore a frame shift mutation. Imagine that a cytosine has been shoved into our sequence, between the first A and G. So now it reads ACGAGUC. Our original 2 codons coded for arginine and valine, but those codons don't exist anymore. The new codon is ACG, and the next is AGU, and these code for the amino acids threonine and serine, respectively. The frame has been shifted down by 1 base. As a result, the last base in the sequence, the last cytosine there on the end, is all by itself. It isn't part of a codon anymore, so it can't code for anything. It's just there, wasted. A second kind of mutation that radically changes the encoded protein is a nonsense mutation. Where a missense mutation creates a protein with a different amino acid, a nonsense mutation creates an artificially short protein. You see, there are 3 codons that are called stop codons, because when they go through the molecular machinery involved in translation, they dislodge the polypeptide. This stops the ribosomes from adding more amino acids, and it creates a free-floating polypeptide chain, which can then fold into a protein. A nonsense mutation happens when an otherwise normal codon is mutated into a stop codon. Where the normal polypeptide should have another amino acid added on, the nonsense mutation causes the protein to be dislodged prematurely. As the protein hasn't been fully constructed, it can't fold properly. The affected protein is basically useless. The chromosomes themselves can also suffer from mutations. I already discussed polyploidy, how having the inappropriate number of copies of a chromosome can lead to serious medical complications like Down syndrome or XXX syndrome. These aren't the only ways that chromosomes can be mutated or disfigured. Segments of chromosomes can be organized or replicated improperly, leading to the impairment of the total chromosome. Sometimes a segment of a chromosome can be totally lost in a deletion event. Other times, a segment may have more copies than it needs, which is called duplication. Recall that during meiosis, chromosomes are deliberately broken at specific points before the crossing over event. Sometimes, accidental breaks in random places can occur. Instead of being cleanly repaired, the broken segments may flip and reattach with the wrong ends. This flip is called a chromosome inversion. In more extreme cases, the broken segments won't even reattach to the other segments of their own chromosome. They'll instead reattach to segments of an entirely different chromosome in a process called chromosome translocation. I know I mentioned cancer a lot in this episode, but never got into too much detail. For those of you who are interested, I'll be talking more about cancer in the future. I plan on having an entire episode dedicated entirely to cancer, where I can talk about what causes it, the different kinds of cancers, how they're treated, all that cool stuff. As for DNA synthesis and repair, I think I've covered all the major points. I hope you were able to learn something cool from this podcast. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.